Photograph a historic site, help Wikipedia, and win a prize. Participate in the world's largest photography competition this month. Learn more. Intermediate filament. 39 languages. Show. Intermediate filament tail domain. Structure of lamina /C globular domain. Identifiers. Symbol if _ tail. FAM PF00932. Interpro IPR 001322. PROSITPTIC 00198. SCOP21IVD /scope /SUPFAM. Available protein structures. Intermediate filament rod domain. Human vimentin coil 2B fragment, CYS2. Identifiers. Symbol filament. FAM PF00038. Interpro IPR 016044. Intermediate filaments, IFs, are cytoskeletal. Structural components found in the cells of vertebrates, and many invertebrates. 1, 2, 3. Homologs of the IF protein have been noted in an invertebrate, the cephalochordate branchiostoma. 4. Intermediate filaments are composed of a family of related proteins sharing common structural and sequence features. Initially designated intermediate because their average diameter, 10 nm, is between those of narrower microfilaments, actin, and wider myosin filaments found in muscle cells. The diameter of intermediate filaments is now commonly compared to actin microfilaments, 7 nm, and microtubules, 25 nm. 1. 5. Animal intermediate filaments are subcategorized into 6 types based on similarities in amino acid sequence and protein structure. 6. Most types are cytoplasmic, but one type, type V, is a nuclear lamin. Unlike microtubules, if distribution in cells shows no good correlation with the distribution of either mitochondria or endoplasmic reticulum. 7. Structure, edit. The structure of proteins that form intermediate filaments, if, was first predicted by computerized analysis of the amino acid sequence of a human epidermal keratin derived from cloned GNAs. 8. Analysis of a second keratin sequence revealed that the two types of keratins share only about 30% amino acid sequence homology but share similar patterns of. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Show. PROSITPTIC 00198. SCOP21GK7 /scope /SUPFAM. Available protein structures. Show. Intermediate filament head, DNA. Binding, region. Identifiers. Symbol filament _head. FAM PF04732. Interpro IPR 006821. SCOP21GK7 /scope /SUPFAM. Available protein structures. Show. Peripherin neuronal intermediate filament protein. Identifiers. Symbol PRPH. Alt. Symbols NEF4. NCBI gene 5630. HGNC 9461. AMIM 170710. Ref seq NM_0062622.3. UNIPROTP 41219. Other data. Locus CHR12Q13.12. Search for show nest in neuronal stem cell. Intermediate filament protein. Identifiers. Symbol NES. NCBI gene 10763. HGNC 7756. AMIM 600915. Ref seek NP_006608. UNIPROTP 48681. Other data. Locus CHR1Q23.1. Search for secondary structure domains. 9. As suggested by the first model, all if proteins appear to have a central alpha helical rod domain that is composed of 4 alpha helical segments, named as 1AM, 1B, 2AM and 2B, separated by 3 linker regions. 9, 10. The central building block of an intermediate filament is a pair of 2 intertwined proteins that is called a coiled coil structure. This name reflects the fact that the structure of each protein is helical, and the intertwined pair is also a helical structure. Structural analysis of a pair of keratins shows that the 2 proteins that form the coiled coil bind by hydrophobic interactions. 11, 12. The charged residues in the central domain do not have a major role in the binding of the pair in the central domain. 11. Cytoplasmic IFs assemble into nonpolar unit length filaments, ULFs. Identical ULFs associate laterally into staggered, antiparallel, soluble tetramers, which associate head-to-tail into protofilaments that pair up laterally into protofibrils, 4 of which wind together into an intermediate filament. 13. Part of the assembly process includes a compaction step, in which ULF tighten and assume a smaller diameter. The reasons for this compaction are not well understood, and if are routinely observed to have diameters ranging between 6 and 12 nm. The N-terminus and the C-terminus of IF proteins are non-alpha helical regions and show wide variation in their lengths and sequences across IF families. The N-terminal head domain binds DNA. 14. Vimentin heads are able to alter nuclear architecture and chromatin distribution, and the liberation of heads by HIV-1 protease may play an important role in HIV-1 associated cytopathogenesis and carcinogenesis. 15. Phosphorylation of the head region can affect filament stability. 16. The head has been shown to interact with the rod domain of the same protein. 17. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Structure of intermediate filament. C-terminal tail domain shows extreme length variation between different IF proteins. 18. The antiparallel orientation of tetramers means that, unlike microtubules and microfilaments, which have a plus end and a minus end, IFs lack polarity and cannot serve as basis for cell motility and intracellular transport. Also, unlike actin or tubulin, intermediate filaments do not contain a binding site for a nucleoside triphosphate. Cytoplasmic IFs do not undergo treadmilling like microtubules and actin fibers, but are dynamic. 19. Biomechanical properties, edit. IFs are rather deformable proteins that can be stretched several times their initial length, 20. The key to facilitate this large deformation is due to their hierarchical structure, which facilitates a cascaded activation of deformation mechanisms at different levels of strain. 12. Initially the coupled alpha helices of unit-length filaments uncoil as they are strained, then as the strain increases they transition into beta sheets, and finally at increased strain the hydrogen bonds between beta sheets slip and the alpha monomers slide along each other. 12. Types, edit. There are about 70 different human genes coding for various intermediate filament proteins. However, different kinds of IFs share basic characteristics. In general, they are all polymers that measure between 9–11 nm in diameter when fully assembled. Animal IFs are subcategorized into 6 types based on similarities in amino acid sequence and protein structure. 6. Types I and II acidic and basic keratins, edit. Further information, cytokeratin. These proteins are the most diverse among IFs and constitute type I, acidic, and type II, basic, IF proteins. The many isoforms are divided in 2 groups. Epithelial keratins, about 20, in epithelial cells, image to right. Trichocytic keratins, about 13, hair keratins, which make up hair, nails, horns and reptilian scales. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Keratin intermediate. Filaments, stained red, around epithelial cells. Vimentin fibers in fibroblasts. Regardless of the group, keratins are either acidic or basic. Acidic and basic keratins bind each other to form acidic-basic heterodimers, and these heterodimers then associate to make a keratin filament. 6. Cytokeratin filaments laterally associate with each other to create a thick bundle of ~50 nm radius. The optimal radius of such bundles is determined by the interplay between the long-range electrostatic repulsion and short-range hydrophobic attraction. 21. Subsequently, these bundles would intersect through junctions to form a dynamic network, spanning the cytoplasm of epithelial cells. Type 3, edit. There are 4 proteins classed as type 3 intermediate filament proteins, which may form homo or heteropolymeric proteins. Dysmin-IFs are structural components of the sarcomeres in muscle cells and connect different cell organelles like the desmosomes with the cytoskeleton. 22. Glial fibrillary acidic protein, GFAP, is found in astrocytes and other glia. Peripherin found in peripheral neurons. Vimentin, the most widely distributed of all IF proteins, can be found in fibroblasts, leukocytes, and blood vessel endothelial cells. They support the cellular membranes, keep some organelles in a fixed place within the cytoplasm, and transmit membrane receptor signals to the nucleus. 6. Syncoilin is an atypical type 3 IF protein. 23. Type 4, edit. Alpha-internexin. Neurofilaments. The type 4 family of intermediate filaments that is found in high concentrations along the axons of vertebrate neurons. Seinman. Syncoilin. Type V nuclear lamins, edit. Lamins. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Lamins are fibrous proteins having structural function in the cell nucleus. In metazoan cells, there are A and B-type lamins, which differ in their length and pi. Human cells have 3 differentially regulated genes. B-type lamins are present in every cell. B-type lamins, lamin B1 and B2, are expressed from the LMNB1 and LMNB2 genes on 5q23 and 19q13, respectively. A-type lamins are only expressed following gastrulation. Lamina and C are the most common A-type lamins and are splice variants of the LMNA gene found at 1q21. These proteins localize to 2 regions of the nuclear compartment, the nuclear lamina, a proteinaceous structure layer subjacent to the inner surface of the nuclear envelope and throughout the nucleoplasm in the nucleoplasmic veil. Comparison of the lamins to vertebrate cytoskeletal IFs shows that lamins have an extra 42 residues, 6 heptads, within coil 1b. The C-terminal tail domain contains a nuclear localization signal, NLS, an Ig-fold-like domain, and in most cases a carboxyterminal CAX box that is isoprenylated and carboxymethylated. Lamin C does not have a CAX box. Lamina is further processed to remove the last 15 amino acids and its furancylated cysteine. During mitosis, lamins are phosphorylated by MPF, which drives the disassembly of the lamina and the nuclear envelope. 6. Type 6, edit. Beaded filaments, phylancin, Farkinin. 6. Nestin, was once proposed for reclassification but due to differences, remains as a type 6 if protein. 24. Vertebrate only. Related to type I4. Used to contain other newly discovered if proteins not yet assigned to a type. 25. Function, edit. Cell adhesion, edit. At the plasma membrane, some keratins or desmin interact with desmosomes, cell-cell adhesion, and hemidesmosomes, cell matrix adhesion, via adapter proteins. Associated proteins, edit. Filaggrin binds to keratin fibers in epidermal cells. Pulctin links vimentin to other vimentin fibers, as well as to microfilaments, microtubules, and myosin II. Kinsin is being researched and is suggested to connect vimentin to tubulin via motor proteins. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Keratin filaments in epithelial cells link to desmosomes. Desmosomes connect the cytoskeleton together, through plakoglobin, desmoplakin, desmoglines, and desmocolins. Desmin filaments are connected in a similar way in heart muscle cells. Diseases arising from mutations in IF genes, edit. Dilated cardiomyopathy, DCM, mutations in the DES gene, 26. Arrhythmogenic cardiomyopathy, ACM, mutations in the DES gene, 27, 28, 29, 30. Restrictive cardiomyopathy, RCM, mutations in the DES gene, 31. Non-compaction cardiomyopathy, mutations in the DES genes, 32, 33. Cardiomyopathy in combination with skeletal myopathy, DES, 34. Epidermolysis bullosa simplex, keratin 5 or keratin 14 mutation. Laminopathies are a family of diseases caused by mutations in nuclear lamins and include Hutchinson-Gilford progeria syndrome and various lipodystrophies and cardiomyopathies, among others. In other organisms, edit. If proteins are universal among animals in the form of a nuclear lamin, the hydra has an additional nematocyllin derived from the lamin. Cytoplasmic IFs, type I4, are only found in bilateria. They also arose from a gene duplication event involving type V nuclear lamin. In addition, a few other diverse types of eukaryotes have lamins, suggesting an early origin of the protein. 25. There was not really a concrete definition of an intermediate filament protein, in the sense that the size or shape-based definition does not cover a monophyletic group. With the inclusion of unusual proteins like the network-forming beaded lamins, type 6, the current classification is moving to a clade containing nuclear lamin and its many descendants, characterized by sequence similarity as well as the exon structure. Functionally similar proteins out of this clade, like crescentins, alveolins, tetrins, and epiplasmines, are therefore only if-like. They likely arose through convergent evolution. 25. References, edit. 1 AB Herman H., Bar H., Kriplick L., Strilkov SV, Ibayu, July 2007. Intermediate filaments, from cell architecture to nanomechanics. Nature Reviews. Molecular Cell Biology. 8, 756-273. Do. 10.1038 / NRM 2197. PMID 17551517. S2 SID 27115011. 2 Chang L., Goldman Road, August 2004. Intermediate filaments mediate cytoskeletal crosstalk. Nature Reviews. Molecular Cell Biology. 5, 860-113 Do. 10.1038 / NRM 1438. PMID 15366704. S2 SID 31835055. 3 Trobe, p. 2012, Intermediate Filaments, a Review, Springer Berlin Heidelberg, p. 33, ISBN 978-3. 642-70230-3. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 4 Carabinosa, Rima D., Erber, Weber K., October 1998. Homologs of vertebrate type I, 2 and 3. Intermediate filament, IF, proteins in an invertebrate, the IF multigeny family of the cephalochordate. Branchiostoma. Feb's letters. 437, 1-15-8 Do. 10.1016 /S0014-5793, 98, 01190-9. PMID 9804163. S2 SID 78863995. 5 Ishikawa H., Biscofa, Holzer H., September 1968. Mitosis and intermediate-sized filaments in developing skeletal muscle. The Journal of Cell Biology. 38, 353855 Do. 10.1083 /JCB.38.3.538. PMC 210837-3. PMID 5664223. 6 ABCDEF Sivni Cassidy H., Chung CW, Lee BT, Common JE, Oges C., It al. March 2008. The Human Intermediate Filament Database, comprehensive information on a gene family involved in many human diseases. Human mutation. 29, 3351-360 Do. 10.1002/HuMu.20652. PMID 180333728. S2 SID 207-60837. 7 Saltis B.J., Gupta R.S., 1992. Interrelationships of endoplasmic reticulum, mitochondria, intermediate filaments, and microtubules. A quadruple fluorescence labeling study. Biochemistry and cell biology. 70, 1011 1174 86 do. 10.1139/092-163. PMID 1363623. 8 Hanukolui, Fuchs C., November 1982. The cDNA sequence of a human epidermal keratin. Divergence of sequence but conservation of structure among intermediate filament proteins. Cell. 31. 124352 do. 10.1016/0092-8674, 82, 90424x. PMID 61863881. S2 SID 35796315. 9 AB Hanukolui, Fuchs C., July 1983. The cDNA sequence of a type 2 cytoskeletal keratin reveals constant and variable structural domains among keratins. Cell. 33, 391524 do. 10.1016/0092. 8674, 83, 90034x. PMID 619181. S2 SID 21490380. 10 Lee CH, Kim MS, Chung BM, Lee DJ, Kolom Pa, June 2012. Structural basis for heteromeric assembly and perinuclear organization of keratin filaments. Nature Structural and Molecular Biology. 19, 770715 do. 10.1038/NSMB.2330. PMC 3864793. PMID 227057888. 11 AB Hanukolui, Ezra L., January 2014. Proteopedia entry, coil structure of keratins. Biochemistry and Molecular Biology Education. 42, 193 do. 10.1002/BMB.20746. PMID 242658184. S2 SID 307-20797. 12 ABC Chin Z, Kriplick L., Buller MJ, October 2009. Hierarchical Structure Controls Nanomechanical Properties of Vimentin Intermediate Filaments. PLOS 1. 4, 10E7294. Bibcode 2009 PLOSO 4.7294Q. Do 10.1371/journal.pone.0007294. PMC 2752800. PMID 198062221. 13 Lodish H., Burko, Zipersky S.L., IT al. 2000. Molecular Cell Biology. New York, W. H. Freeman. P. Section 19.6, Intermediate Filaments. ISBN 978-0-07-243940-3. 14 Wang Q, Tolstonog G.V., Schumann R., Trobe P., August 2001. Sites of nucleic acid binding in type I4. Intermediate filament subunit proteins. Biochemistry. 40, 34103429 do, 10.1021/by01083005. PMID 115136113. 15 Schumann RL, Hutterman C, Hartig R, Trobe P, January 2001. Amino-terminal polypeptides of vimentin are responsible for the changes in nuclear architecture associated with human immunodeficiency virus type 1 protease activity in tissue culture cells. Molecular biology of the cell. 12, 114354 do, 10.1091/MBC.12.1.143. PMC 30574. PMID 11160829. 16 takes Mura M, Gami H, Coloxygione, Itahara S, August 2002. Protective role of phosphorylation in turnover of glial fibrillary acidic protein in mice. The Journal of Neuroscience. 22, 16 6972 9. Do. 10.1523/22-16-06972.2002. PMC 6757867. PMID 121779195. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 17 Parry the, Merkofel N, Steinert PM, Smith Ta, 2002. A role for the 1AM and L1 rod domain segments. In head domain organization and function of intermediate filaments, structural analysis of trichocyte. Keratin. Journal of Structural Biology. 137, 1297-108 do, 10.1006/sp.2002.4437. PMID 12064937. 18 Quinlan R, Hutchison C, Lane B, 1995. Intermediate filament proteins. Protein profile. 2, 8795. 952 PMID 8771189. 19 Helfand BT, Chang L, Goldman Road, January 2004. Intermediate filaments are dynamic and motile. Elements of cellular architecture. Journal of Cell Science. 117, pt 213341. Do. 10.1242/JCS.00936. PMID 146762669. 20 Herman H, Bar H, Kriplik L, Strilkov SV, Ibayu, July 2007. Intermediate filaments, from cell architecture to nanomechanics. Nature Reviews. Molecular Cell Biology. 8, 756273. Do. 10.1038/NRM 2197. PMID 17551517. S2 SID 27115011. Z, Kriplik L, Buller MJ, October 2009. Hierarchical structure controls nanomechanical properties of vimentin intermediate filaments. PLOS 1. 4, 10E7294. Bibcode, 2009 PLOSO, 4.7294Q. Do. 10.1394/journal.pwn.0007294. PMC 2752800. PMID 198062221. Fudge D. January 2007. Biomechanical properties of intermediate filaments, from tissues to single filaments and back. Bioessays. 29, 12635. Do. 10.1002/bis.20514. PMID 171873757. S2 SID 6560740. Chin Z. Buller MJ, Krippler L. January 2010. A multiscale approach to understand the mechanobiology of intermediate filaments. Journal of Biomechanics. 43, 11522. Do. 10.1016/j.biomic.2009.09.004. PMID 198117883. Chin Z. Krippler L. Buller MJ, October 2009. Nanomechanical properties of vimentin intermediate filament. Dymus. Nanotechnology. 20, 42 42425101. Bibcode 2009 Nano 20p 5101Q. Do. 10.1088/0957. 4484/20/42/4255101. PMID 1977920. S2 SID 6870454. 21 Haymufi, Windorfer A, Alubri, Urbach M, Kozloff mm, July 2020. Model for bundling of keratin. Intermediate filaments. Biophysical Journal. 119, 16574. Bibcode 2020 BPJ. 119, 65H. Do. 10.1016/j.bpj.2020.05.024. PMC 7335914. PMID 325333940. 22 Brodiller, Gayaneramala, Milting H., August 2018. Molecular insights into cardiomyopathies associated with dysmin, DES, mutations. Biophysical Reviews. 10.4983/1006. Do. 10.1007/S12551-018-0429-0. PMC 6082305. PMID 299264427. 23 Zinc oil in Homo sapiens, human, zinc gene and protein. www.unipred.org. Retrieved. 20th of December, 2021. 24 Bernala, Arans L., June, 2018. Nestin expressing progenitor cells, function, identity and therapeutic implications. Cellular and Molecular Life Sciences. 75, 12 2177 2195 Do. 10.1007/S00018-018. 2794 Z. PMC 59480302. PMID 295411793. 25 ABC Colmar M., May 2015. Polyphily of nuclear lamin genes indicates an early eukaryotic origin of the metazoan type intermediate filament proteins. Scientific reports. 5. 10652. Bibcode 2015 NAT SR 510652K. Do 10.1038/SREP10652. PMC 444859. PMID 260240016. 26 Fisher B, Dittman S, Brodiller, Unger, Stallmeyer B, Paul M, et al. December 2020. Functional characterization of novel alpha helical rod domain dysmin, DES, pathogenic variants associated with dilated cardiomyopathy, atrioventricular block and a risk for sudden cardiac death. International Journal of Cardiology. 329, 167174. Do 10.1016/j.icard.2020.12.050. PMID 33373648. S2 SID 2297198883. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 27 Bermudez Jimenez FJ, Carriol V, Brodiller, Alaminos M, Camposo, Sciarama I, et al. April 2018. Novel dysmin mutation P.Glu401 ASP impairs filament formation, disrupts cell membrane integrity, and causes severe arrhythmogenic left ventricular cardiomyopathy/dysplasia. Circulation 137. 15 1595 1610 do 10.1161/circulation AHA.117.028719 HDL 10481/89514. PMID 29212896. S2 SID 4715358. 28 Protonotriosa, Brodiller, Asamacchio, Jagger J, Quinney, Stanaziuk C, et al. December 2020. The novel dysmin variant P.Lu115L is associated with a unique form of biventricular arrhythmogenic cardiomyopathy. The Canadian Journal of Cardiology. 37, 6857866. Do 10.1016/j.kchka.2020.11.017. PMID 33290826. S2 SID 2280786848. 29 Clark B, Cosman S, Gayener, Brand K, Stork I, Brodiller, et al. December 2010. De novo. Dysmin mutation N116S is associated with arrhythmogenic right ventricular cardiomyopathy. Human molecular genetics. 19, 23 4595 607 do 10.1093/HMG/DDQ387. PMID 20829228. 30 Brodiller, Hedi PN, Diding M, Fatima, Walhorn V, Gader S, et al. May 2012. Dual color photoactivation localization microscopy of cardiomyopathy-associated dysmin mutants. The Journal of Biological Chemistry. 287, 1916044757 do 10.1074/JBC.M111.313841. PMC 33461104. PMID 22403400. 31 Karat Brodilla, Por Hakimi Sat, Stanazi Uksi, Ratnave Diveles, Hendig D, Gayaner, it al. November 2019. Restrictive cardiomyopathy is caused by a novel homozygous dysmin, DES, mutation. P.Y122H leading to a severe filament assembly defect. Genes 10, 11 918. Do 10.3390/genes10110918. PMC 6896098. PMID 317180026. 32 Karat Clera, Helen Broik Y, Van der Ven PF, First Do, Hubner, Bruchert Cipher V, it al. December 2007. Clinical and morphological phenotype of the filament myopathy, a study of 31 German patients. Brain, a Journal of Neurology. 130, pt 12 3250 64 do, 10.1093/brain/orm 271. PMID 180555494. 33 Karat Marek Honof Brodilla, Musnikov RP, Sparba Pa, Kaislava AV, Kulikova it al. June 2019. Non-compaction cardiomyopathy is caused by a novel in-frame dysmin, DES, deletion mutation within the 1am coil rod segment leading to a severe filament assembly defect. Human mutation 40. 6734741 10.1002/HuMu.23747. PMID 309089796. S2 SID 855155283. 34 Carats Chiarama I, Diding M, Klauk B, Brodilla, Gayaneramala, Walhorn V, it al. March 2018. A novel dysmin, DES, indel mutation causes severe atypical cardiomyopathy in combination with atrioventricular block and skeletal myopathy. Molecular Genetics and Genomic Medicine. 6, 2288. 293 do, 10.1002/MGG3.358. PMC 59022401. PMID 29274115. Further reading, edit. Herman H. Harris Jr., eds. 1998. Intermediate filaments. Springer. ISBN 978-0-306-45854-5. Omai MB, Colompa, eds. 2004. Intermediate filament cytoskeleton. Gulf Professional Publishing. ISBN 978-0-12-56417-3-9. Paramayo J.M., ed. 2006. Intermediate filaments. Springer. ISBN 978-0-387-33780-7. External links, edit. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Intermediate and filament and proteins at the U.S. National Library of Medicine Medical Subject. Headings, Mesh. This article incorporates text from the public domain FAM and Interpro, IPR 001322. This article incorporates text from the public domain FAM and Interpro, IPR 006821. Categories, Genes on human chromosome 12, genes on human chromosome 1. Protein families cytoskeleton. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF.